So today with this video, you're going to learn all of the steps of glycolysis, you're going to learn how it works, and also you're going to understand how to memorize it and truly comprehend it. And at the end of the video, we're going to create this entire map together. And you'll also just have all of the flashcards and this map if you want to use it, but I'd really recommend that you create your own map and really learn how to make this yourself. But hopefully this gives you an idea of how you can study and learn glycolysis in a way that will help you. Before we start, I just want to talk about why it's so helpful and important to learn glycolysis. You know, why are your teachers making you learn this? It's because teachers know that glycolysis is a great way to understand all of the different biochemical reactions in your body. It uses a lot of common enzymes that you're going to learn, like hexokinase or kinases, that are used throughout the body. And that's why you're learning glycolysis. It's kind of like an example or a model to show you how future, more complex mechanisms work. So with that, let's jump into glycolysis. Okay, so let's start with glucose. You consume glucose, it goes through your digestive tract, and then it's going to end up in the bloodstream. And then it's going to come into contact, for example, with a muscle cell. This is one place where glycolysis occurs. So glucose is going to enter the muscle cell, it's going to enter the cytosol, and then it's going to enter into glycolysis. Glycolysis isn't a place, it's just a process that occurs within the cytosol. So glycolysis, the moment of truth. So glycolysis is really cool. It's basically a waterfall or pathway of different enzymes that will convert glucose into the final product, which is pyruvate, and create other things that are useful for the body in the process. So let's see how that happens. So first, glucose is going to come in contact with an enzyme. You're going to see lots of enzymes in this. The first enzyme is hexokinase. When you see the word kinase, you're going to think of phosphate. Kinases are a group of enzymes that basically add phosphates. So we're going to put this underneath, this glucose 6-phosphate. Let me zoom in a little bit here. So basically we've got our glucose, and then it's going to come down and it's going to go to hexokinase, and then this enzyme is going to convert it into glucose 6-phosphate. So do you see how we've got this phosphate added to where this alcohol group was? So that's what that hexokinase did. Now you might be wondering, where on earth did that ATP come from? So where on earth did that phosphate come from? I got ahead of myself there, but it came from ATP. So ATP has phosphate in it. So the ATP comes in here, the hexokinase uses it to create that phosphate, and in a byproduct of that is going to be ADP. So we have a triphosphate that converted to a diphosphate, and this extra phosphate was added to the glucose. So that's what hexokinase does. So I'm just going to quickly write that again here. The kinases adds phosphates. And once again, you'll have access to all of these notes and flashcards afterwards. But let's go on to the second step. So we've got glucose 6-phosphate, and then I'm going to bring this down. This is the next product that we're going to see. It's called fructose 6-phosphate, okay? Glucose, fructose, maybe you've heard of that. You can see how the structure's changed a little bit. So what type of enzyme would do that? Well, it's an enzyme called an isomerase. Isomerase, when you hear of isomerases, that's a type of enzyme that is going to change, kind of mold or restructure a part of a molecule. So let's put in here, this is going to be phosphoglucose isomerase. See, we've got that phosphoglucose right here, and now we're changing it. It's an isomer. So once again, we've got kinase, which adds phosphates, and then isomerase, which kind of changes the molecule. So let's add that in here. So now this glucose is going to enter this enzyme. This enzyme is going to create then fructose 6-phosphate. Once again, remember that we're learning these enzymes not just for glycolysis, but to understand these typical enzymes that happen within your body. You're going to see lots of isomerases and kinases in your body. Okay, let's keep going. So what happens after this? Well, after fructose 6-phosphate, we are going to add, let's see the next product, fructose 1,6-biphosphate. So you might think, okay, so we're adding another phosphate on here. How or what type of enzyme would do that? And if you thought of a kinase, then you were right on. It's going to be phosphofructose kinase 1. Phosphofructose kinase 1. I'm not the best speller, so hopefully that's right on. But once again, kind of like this hexokinase, right, where we're adding a phosphate, the same thing's going to happen here. So let's add a phosphate to do this. Let me just link this up here. So that's going to enter here, and then it's going to add another phosphate. Awesome. Okay, and when we add phosphates, what do we need again? Oh, we're going to need that ATP again. So let me just create some ATP real fast. So when you're seeing kinase now, you're thinking, okay, kinase is what's going to add phosphates to a molecule. And then whenever we're going to be using ATP, you can just assume and know that really ADP is going to be produced in the process. So let's add that here. Okay. We're doing great. So you've already learned what a kinase is, what an isomerase is. So let's keep going. So what's the next step that we're going to learn? Let's go up here to our next molecule. We're going to see kind of some weird ones here. We've got a DAP, whatever that is, and a GAP. So you're like, oh my goodness, what are these? So let's talk about it. We go through this molecule, and this molecule has to turn in to these kind of two strange molecules. And what would cause that? So to create that, we're actually going to use something called an aldolase. And this isn't maybe a common enzyme like kinases or isomerases. It's an enzyme that specifically causes this type of reaction. So anyway, so we have the fructose 1,6-biphosphate come in, and then this is going to break out into these two. Whew. And we're almost there. So now, once we do this, the body's going to favor this GAP for the next process. So we just want to convert this DAP into that. And to do that, how do we change a simple structure into a similar simple structure? What kind of enzyme does that? You might have guessed it, it's an isomerase again. So we're going to use an enzyme called triose phosphate isomerase. Okay, so this triose phosphate isomerase is going to convert this DAP into GAP. And that ends the first phase of glycolysis. So GAP here, now we actually, even though it's showing just one, we actually have two GAP molecules. So for the second phase of glycolysis, it's important to know that this is happening 2 times. So GAP needs to be converted. Let's take this up here for a second. GAP needs to be converted into 1,3-BPG. And to do that, we're actually going to have kind of a cool reaction. Let me kind of zoom in here. So we're going to use, and remember this is happening twice starting from here. To do that, we're going to use an enzyme called GAP or G3P dehydrogenase. And to do this, we're going to need a coenzyme that you're going to use a lot and see a lot in metabolism, and that's called NAD+. And then, kind of like ATP turning into ADP, this NAD is going to steal a hydrogen, so it's going to turn into NADH. So this is that next step right here. Once again, this is all happening twice, so let me just kind of add that here. So the two gaps are going to be converted into two of these 1,3-BPGs, and to do that, we're going to need NAD. Okay, so what else is going to happen? So the next step of this process is getting rid of one of these phosphates. And so with everything you've learned so far, what enzyme might be involved with that? Well, if you're thinking of a kinase, you're right on. So let's add that in here. So we're going to add one called phosphoglycerate kinase. So let's add that in here. 1,3-BPG adds into there. It's going to be into the glycerate. And then what's going to happen to this? If you see this extra phosphate, where is it going to go? If you thought of ATP, you're right on. We're going to create ATP. And I'm just going to write this out here. We're actually creating 2 ATP if you're thinking about it, because this whole process is happening twice. So we can go in here, and that's going to come from 2 ADP. You can kind of already tell that these ATPs and ADPs, they normally kind of get converted into one another. So it's going to enter into here. This phosphorate glycerate kinase uses it to create this 3-phosphoglycerate. Now, what happens next? So now let's look at the next reaction. So we're going to need to create 2-phosphoglycerate. And how on earth are we going to do that? Well, it changes a little bit of the structure. So let's look at how that happens. If you're thinking of an isomerase, you're really thinking right on. This one's not going to say isomerase. It's going to have a special name called a mutase, but it's basically doing the same function. So we're going to add the enzyme phosphoglycerate mutase, and that is what converts 3-phosphoglycerate into phosphoglycerate mutase into 2-phosphoglycerate. Sorry, this product right here isn't getting converted into this. This is the enzyme. So we've got 2-phosphoglycerate. If you're staying with me here, we're almost done. Then what's going to happen after that? So then we have to create something called PEP. And let's kind of see how that works. Let me just kind of expand this a little bit so we've got a little bit more room here. So we've got to create this PEP, and the enzyme that's going to make this is called an enolase. You might have learned about enols in your organic chemistry class if you've already taken that. So we're going to use an enolase to convert this into here. And then we've got PEP, and we're so close. Then the last one we need is we need to convert this PEP into pyruvate. And this right here, this molecule right here, is called pyruvate. And this is an important molecule. This is the last molecule created from glycolysis. Let me just add this into here. Okay, so we've got pyruvate, we've got PEP, and what's the last enzyme? So if you've noticed here, it kind of looks like we're going to be losing maybe a phosphate. I wonder what would do that. What enzyme would do that? Would that be an isomerase or a kinase? If you're thinking about it, once again, those kinases are what changes and adds and subtracts those phosphates. So this is going to be pyruvate kinase. Sorry, I was doing bad with the spelling there. So we've got pyruvate kinase, and once again, kind of like you've already seen, we need ADP and ATP to make that happen. So let's go like this. ADP. That's going to enter into here. And then ATP. And this is actually creating 2 ATP. Remember, the second part of the cycle is all happening twice. So that's just kind of an important thing to remember. So, you've done it. That's the second stage of glycolysis. Let's just kind of look over this. So we've got this first stage, right? We've got glucose. It gets converted all the way down, if you can see it, into this gap. We're going to be using ATP in that whole process. And then up here, we go through the second stage, which happens twice. And we're actually going to be creating 4 ATP. So there's 2 ATP there. Remember, this whole process is happening twice, so I can just do 2 ATP here. So in total, we're going to be making 2 ATP from this process because we needed to use 2 ATP here, and then we were creating 4 ATP over here in the second half of it. So that's how that works. Once again, big picture, why are you studying glycolysis? Glycolysis is helping you learn important enzymes that will be used throughout the body, or types of enzymes. Let me put this in the cytosol. Remember, glycolysis happens within the cytosol, and it's all about breaking down glucose. So hopefully that explains all of that for you. And once again, these flashcards and the notes will all be available for you if you need them. Best of luck. Let me know if you have any questions or if you saw any mistakes.